Real-time PCR, like PCR in general, is used to amplify regions of interest in the nucleic acid so that a region of DNA can be detected or quantified if required. As quantification is possible, real-time PCR is also called quantitative PCR. Real-time PCR amplifies DNA based on the same principle as other types of PCR, that is, cycles of denaturation, annealing, and extension. In real-time PCR, after each cycle of reaction progress, amplification is monitored using fluorescence. Fluorescence is generated because the basic PCR mix, consisting of DNA template, primers, deoxynucleotides, DNA polymerase, buffer, and cofactor, is added with, for example, 1. A fluorescent dye that non-specifically intercalates with only double-stranded DNA, or 2. Sequence-specific DNA probes having fluorescently labeled reporters at the 5' end and quenchers at the 3' end. These probes bind to the amplified region, and when the probe's fluorophore is separated from the quencher by the nuclease activity of Taq polymerase as it synthesizes a new DNA strand, the reporter generates fluorescence. Probe chemistry allows multiplex reactions in a single tube or plate well, using probes with unique dyes for each reaction. As DNA is amplified, there is more intercalation of dyes or more probes with reporters separated from quenchers, so more fluorescence is detected when the dyes are excited at a specific wavelength of light from the real-time PCR instrument or thermocycler. Fluorescence intensity measurements are reported as relative fluorescent units, RFU, as fluorescence intensity depends on many factors, such as the instrument setting, detector sensitivity, and optical system. Some PCR systems report the normalized reporter value, Rn, to correct for well-to-well differences in fluorescence. Rn is obtained by dividing the fluorescence emission intensity of the reporter dye, R, by the fluorescence emission intensity of a passive reference dye, P. Fluorescence, or normalized reporter fluorescence, plotted against the cycle number, results in an amplification curve, typically sigmoidal, with 4 distinct phases: lag, exponential, linear, and plateau. During the lag phase, amplification occurs, but the signal is not strong enough to be detected above the background. The background may be from, for example, the remaining probe fluorescence that is not quenched and light leaking into the sample well. During the exponential phase, the amount of PCR product approximately doubles in each cycle as the reaction mix contains abundant reaction components. During the linear phase, the reaction slows down as reagents run low. During the plateau phase, no change occurs as one or more components may be limiting. The amplification curve may be represented by a logarithmic y-axis scale, which provides a more proportional representation of the data. For example, on a linear scale, lower expressed data points would appear much closer together than higher expressed data points, even though the fold difference between the points is the same. The logarithmic scale makes it easier to identify the exponential phase of the amplification curve. Important parameters in the amplification curve include the baseline, threshold, and quantification cycle, or CQ. The baseline is the level of fluorescence typically measured between cycles 3 and 15, which has low amounts of amplification products, so much of the fluorescence is noise or background. The threshold is the level of signal that reflects a statistically significant increase over the baseline signal. The quantification cycle or CQ, sometimes called the threshold cycle or CT, corresponds to the PCR cycle in which the sample amplification curve crosses the threshold level. CQ is critical for determining the initial amount of amplified product. So for accurate determination of the CQ, the normalized and baseline corrected fluorescence delta Rn is obtained by subtracting the average fluorescence value detected within the early cycles from the fluorescence value obtained from amplification products. As seen here, the precision of replicates can increase with the use of delta Rn. Generally, the instrument software sets the baseline, calculates delta Rn, and sets the threshold value. However, sometimes settings have to be manually modified. Reliable quantification of results also depends on assay, efficiency, specificity, sensitivity, precision. So these parameters should be examined. Efficiency of PCR, that is, whether the DNA is actually doubling every cycle, is indicated by amplification curves. A shallow curve indicates poor efficiency, while a steeper rise indicates better efficiency and sensitivity. One way to measure efficiency is to generate a standard curve by running serial dilutions of DNA template, which may be of known concentration or a sample of unknown concentration. CQ values obtained for each dilution is plotted against the log of the starting concentration of the template or the dilution factor of unknown concentration. Efficiency is then calculated using the equation E is equal to 10 to the power of negative 1 divided by the slope minus 1, so that the maximum efficiency is 1 rather than the 2 due to doubling. Percentage efficiency is equal to E times 100. Efficiencies under 85% or over 100% are not recommended for data analysis, and you should optimize the PCR reaction by modifying the components and/or the conditions of the qPCR assay. Specificity refers to whether the fluorescent measurement is only from the gene of interest and can be determined by using the dissociation or melt curve, amplicon size, and the sequence of a PCR product. In melt curve analysis, temperature is gradually increased, so DNA melts or denatures and fluorescence decreases. A clear view of melting dynamics is seen when negative change in fluorescence divided by change in temperature is plotted against temperature. A distinct melt peak typically characterizes specific amplification, while multiple melt peaks will likely be present if multiple PCR products are formed due to non-specific amplification. Confirming the validity of results also requires controls. The no-template control, NTC, tracks contamination or primer-dimer formation. A negative or no amplification control, which has no DNA polymerase, tracks the reaction's background signal and probe stability. An endogenous positive control is used to correct for quantity and quality differences between samples and uses a reference gene whose expression does not vary for the treatments investigated. Usually, it is a housekeeping gene constitutive in expression. An exogenous positive control is used to test for the presence of inhibitors or test master mix performance by using an unrelated sequence spiked into the samples at a known copy number. Precision or consistency of measurement contributes to the accuracy of quantification in PCR and is measured using technical replicates, that is, replicates from the same sample. Biological replicates from different individuals or specimens are required to draw biologically relevant conclusions from your experiment. Once the PCR assay is validated and it is confirmed that the fluorescence measured is due to the gene of interest, the next step may be to quantify the initial concentration of the locus of interest. Depending on the experimental question, quantification may be absolute in reference to an appropriate standard curve or relative, a comparison between samples. For absolute quantification, an absolute standard curve is created by diluting a nucleic acid sequence that has been accurately quantified by other means. The DNA quantity of target or gene of interest in the sample is obtained by comparing the CQ values of the unknown targets to the standards. Using the straight line equation, CQ equals m times log of quantity plus b, where m is the slope and b is the intercept on the y-axis, the DNA quantity of the target can be calculated and is equal to 10 to the power of CQ minus b divided by m. Sample concentration should only be determined in the linear portion of the standard curve. There should be no extrapolation of the straight line to determine the concentration of the sample, as beyond the range tested, the relationship between CQ and log of dilution may not be linear. Concentration of the gene of interest in the sample has to be normalized for valid comparisons. Consider this example. Without normalization, sample A appears to have more DNA than sample B in the 1 ml extracts. However, once normalized for mass of tissue, sample B has a higher concentration of DNA than sample A. The normalizer may be, say, the amount of tissue or the number of cells used for template preparation, microgram of nucleic acid used as PCR template, or the expression level of a reference gene. In the case of relative quantification, the expression of a gene of interest in the sample, example a treated sample, is compared to that in another sample, or control or calibrator, example an untreated sample. A normalizer or reference gene, such as GAPDH, is used as a control for experimental variability. The results are expressed as fold change increase or decrease in the treated sample to the untreated sample. For example, sample A has threefold higher or 3 times as much DNA as sample B. There are a number of ways of calculating the fold difference, each with its benefits, drawbacks, and limitations. For example, if there are differences in PCR efficiencies between the gene of interest and the reference gene, PCR efficiencies of the gene of interest and the reference gene have to be taken into account when calculating the relative expression ratio R of the target or gene of interest to the reference gene. Thus, R is equal to 1 plus the efficiency of the gene of interest to the power of delta CQ of the gene of interest, all divided by 1 plus efficiency of reference gene to the power of delta CQ of the reference gene. Efficiency is equal to 10 to the power of negative 1 over S minus 1, where S, the slope, is obtained from the graph of CQ versus log of concentration. Delta CQ is equal to CQ of the calibrator or control minus the CQ of the unknown or test sample. If there are no differences in PCR efficiencies and PCR efficiencies are 100%, the fold change can be calculated using the delta-delta CQ method. To do so, first normalize the CQ of the gene of interest to the CQ of the reference gene for both control and test sample using the equation delta CQ equals CQ of the gene of interest minus CQ of the reference gene. Then normalize the delta CQ of the test sample to the delta CQ of the control. Using the equation delta delta CQ is equal to delta CQ of the unknown or test sample minus delta CQ of the control. And finally, calculate R using the equation R equals 2 to the negative power of delta delta CQ. The PCR system software should provide you with the graphs and calculated values required. So in summary, 1. Real-time PCR is used to amplify regions of interest in nucleic acids so that a region of DNA can be detected and, if required, quantified. 2. Real-time PCR assays efficiency, specificity, reliability, accuracy, and sensitivity should be checked to obtain valid data. If necessary, optimize the reaction. Also, normalize values. 3. Real-time PCR quantification of starting quantities of DNA may be absolute, example X copies of DNA per unit volume per gram of sample, or relative, for example, the sample has X-fold more DNA than the control. Now let's review with questions and answers. Question 1. Assume you conduct a real-time PCR experiment using A 0.1 nanogram, B 0.5 nanogram, and C 1.45 nanogram of the same template DNA. Do you expect the threshold cycle to decrease or increase when going from A to B to C? Answer: CQ should decrease going from A to B to C, as the higher the initial quantity of DNA, the faster it will reach a specific amount when doubling. Question 2. A virus, when present in human blood, causes sickness. In testing drug treatments, blood of equal amounts taken from an individual before treatment, A, and after treatment, B, were analyzed. The graph shows the amplification curves of gene X from the virus. What happened to gene X in the treated cells? A, its copy number increased. B, its copy number decreased. C, the copy number did not change. D, no conclusion can be made. The answer is B. Detectable fluorescence starts about 5 cycles earlier in sample A than in sample B, indicating that gene X is more concentrated in the patient's blood before treatment than after treatment. Question 3. Why does fluorescence not increase after cycle 30 in either sample that underwent real-time PCR using Tachman probe-based chemistry? A, because all primer molecules have been used up. B, because all Tachman probe molecules have been used up. C, because all template molecules have been used up. D, A or B. E, A, B, and C. The answer is D. The quantity of primers and probes in the PCR mix depends on how much is added during preparation of the mix and will be used up. However, the template increases every round of amplification. Question 4. What is one benefit of performing baseline subtraction? A, ensures the amplification efficiency is 100%. B, improves specificity. C, improves precision of technical replicates. D, all of the above. The answer is C, improves the precision of technical replicates. Question 5. Select the human error or errors that reduce the accuracy of real-time PCR results. A, incorrect data analysis. B, improper assay development. C, improper experimental design. D, all of the above. The answer is D, all of the above. If you found this video useful, support the channel by subscribing and sharing with your friends and colleagues. Thank you.